close your eyes for a moment. Actually, wait. Before you close them, I want you to do something. Something so simple it will feel almost foolish. And yet, by the end of this night, this long, slow, starlit journey we are about to take together, you will understand why I am asking. Look at your hand. Go ahead. Hold it up if you can. Or simply feel it where it rests. Against the blanket, against your chest, pressed lightly into the softness of your pillow. Feel the warmth radiating from your palm. Feel the weight of your fingers. Notice, if you can in the low light, the faint blue threads of veins tracing their way beneath your skin like rivers seen from very high above. That hand, that specific hand, with its particular fingerprints and its small history of scars and its joints that ache slightly on cold mornings. That hand is the end result of a chain of survival so improbable, so achingly long, so filled with cold nights and close calls and impossible decisions, that mathematicians would struggle to calculate its odds. And somewhere inside that hand, inside the red rivers running beneath the thin geography of your skin, there's a molecule, a tiny protein, a microscopic flag planted on the surface of your red blood cells, so small that even the most powerful electron microscopes must work to resolve its shape. That protein has a name. We call it the rhesus factor, the Rh antigen, and whether it is present on your blood cells, or startlingly, mysteriously, defiantly absent, tells a story that reaches back further than civilization, further than agriculture, further even than the oldest cave paintings in the dark chambers of southern Europe. It reaches, perhaps, to a divergence so ancient and so strange that scientists still argue in hushed academic tones about what it truly means. Tonight, we're going to follow that river together. Slowly. There is no rush here. This is not a lecture hall. This is not an exam. Think of this instead as a campfire, a very old one, burning in a very dark place. And we are all gathered around it together. Right now, as these words settle into your ears and drift towards sleep, there are listeners in Osaka and Oslo, in Lagos and Lima, in small apartments in Istanbul, and large houses in the English countryside, all lying in the same soft dark, all breathing the same slow breath, all holding, whether they know it yet or not, the same ancient mystery inside their veins. You are not alone in this dark, and you do not need to remember any of this. That is the first permission I want to give you tonight, and I will give it many times before the last scene fades like footsteps on a tide-washed shore. You do not need to hold onto the names. You do not need to catalog the dates or memorize the science. Let the details come to you like weather, like the particular smell of rain approaching across a field you can't quite see, and then let them pass through you and dissolve into the warm dark behind your closed eyes. Just listen. Listen the way our ancestors listened around their fires. Not analytically, not with the sharp, acquisitive attention of a student taking notes, but with the older, softer, more animal kind of listening, the kind where the story lives in your body before it lives in your mind. Now, let me tell you about the blood. About 15% of human beings walking the earth today, roughly 1 in every 7 people, carry something in their blood that the other 85% do not. Or more precisely, they carry the absence of something, a missing protein, a gap in the molecular landscape of their red blood cells where a particular antigen should, by all evolutionary logic, exist. We call these people Rh negative. And for decades, for generations of researchers staring at petri dishes and population maps with deeply furrowed brows, the existence of these people has presented biology with something almost embarrassing, a mystery it cannot fully solve. Because here is the paradox that will slowly unfold across this entire night's journey like a long, slow tide pulling back to reveal a strange and ancient shore. Evolutionary biology does not typically preserve traits that cause harm. It eliminates them, quietly, efficiently, over thousands of generations, like a slow and patient editor removing unnecessary sentences from a very long book. And yet, Rh negativity causes harm. Real, measurable, physiological harm. Specifically, it causes a condition during pregnancy called hemolytic disease of the newborn. When an Rh-negative mother carries an Rh-positive baby, which, given that Rh-positive is dominant, happens frequently, her immune system can, under certain circumstances, learn to recognize her own child as a foreign invader and attack it. This is not metaphor. This is molecular warfare between a mother and her unborn child. Evolution should have weeded this out long ago. In the cold mathematics of natural selection, a trait that causes mothers to attack their own children should have been quietly, efficiently removed from the human story. And yet, here we are, 15% of us, scattered across the globe in a pattern that is not random, that clusters in strange ways in certain populations, that appears in higher concentrations in some of the most ancient, isolated communities on Earth, and in lower concentrations in others. A 15% slice of humanity carrying a molecular mystery that science has not, despite its best and most rigorous efforts, fully explained. That is where we begin tonight, with your hand in the dark, with the blood moving quietly beneath your skin, with the ancient, unresolved question humming in the very cells of your body. Let your breathing slow now. Let the weight of the blanket settle over you like a comfortable history. Let your shoulders drop. Let your jaw unclench. We have a long night ahead. A beautiful, slow, starlit journey through deep time. And there is nowhere else you need to be. It is 1937. The world is moving quickly in terrible directions. The newspapers are full of dark winds blowing from Europe, and the sky over most of the continent has that particular gray quality of impending weather. But in a laboratory in New York City, two scientists are doing something quiet and extraordinary. They are not building weapons or formulating theories of power. They are simply looking, very carefully, at blood. Their names are Carl Landsteiner and Alexander Wiener. Landsteiner, at this point, is already legendary, a Nobel laureate, an Austrian physician who decades earlier had discovered the ABO blood group system, and, in doing so, saved countless lives by making blood transfusions safe for the first time in medical history. He is old now, white-haired, meticulous, with a particular kind of patience that belongs to men who have spent their lives looking at things most people cannot see. And in 1937, working with the blood of rhesus macaque monkeys, those particular, dark-eyed primates with their expressive faces and their ancient evolutionary relationship to us, Landsteiner and Wiener discover something new. They find an antigen, a protein, present on the red blood cells of the rhesus monkeys, and when they test human blood against antibodies produced from this protein, present in the blood of most, but not all, humans too, they call it the rhesus factor, Rh, a nod to the monkeys, a scientific shorthand that will carry, embedded within it, the first seeds of a profound misunderstanding. Because here is what the naming implies, and what would, for years, send researchers down a gently wrong path. The name rhesus factor suggests a connection, a shared inheritance between humans and rhesus monkeys, as though Rh-positive humans are slightly more monkey, as though Rh-negative humans are something else, something more diverged, something whose lineage forked away from the standard primate path at some important, unspecified point in the distant past. This implication, never stated plainly, always hovering just beneath the surface of the science, would feed decades of speculation, some of it thoughtful, some of it careful and rigorous, and some of it, frankly wild, conspiratorial, reaching into territory that science does not and cannot support. But the mystery it points toward is real. The mystery is genuine. Let the warmth of your bed hold you for a moment as we let that sink in. The discovery is made in 1937 in a quiet New York laboratory, and from that moment forward, biology has been left with an uncomfortable question sitting at its foundations like a stone that doesn't quite fit. Landsteiner and Wiener don't know, in 1937, about the full complexity of what they found. The Rh blood group system, as science would later reveal, is not a single antigen, but a family of proteins encoded by two closely linked genes on chromosome 1, the RHD gene and the RHC gene. The Rh-positive phenotype results from the presence of the RHD antigen on red blood cells. Rh-negative individuals simply do not express this protein, in most cases, because they have inherited two non-functional copies of the RHD gene. The gene, essentially, is silent, or deleted, or quietly absent. Let that image sit with you gently. Not a mutation that added something strange, but an absence, a silence where something used to be, or where something, in everyone else, still exists. Picture a symphony playing in a great stone hall. Every instrument present, the sound enormous and complete. And then imagine that in one section of the audience, 1 in 7 people, certain instruments simply are not playing. The music continues. It is not broken, but there is a particular quality of quiet in that corner of the hall that is not the same as the rest. That is Rh negativity in the rough poetry of metaphor. An absence, a silence, a gap. But here's where the science becomes immediately, compellingly strange. When Landsteiner and Wiener begin mapping this factor across human populations, they find something that should make any evolutionary biologist sit up very straight in their chair. The distribution is not random. Rh-negative blood does not appear with equal frequency everywhere humans live. It clusters. It concentrates. It forms geographic patterns that map, with uncomfortable precision, onto some of the oldest, most isolated, most genetically distinctive human populations on Earth. Specifically, among the Basque people of the Pyrenean Mountains between France and Spain, Rh-negative frequency reaches 30 to 35%, sometimes higher. In some isolated Basque villages, the percentage climbs even further, 35%, in a world where the global average is approximately 15%, more than double. The Basque people, it should be noted, are already one of the great mysteries of European anthropology. Their language, Euskera, is a linguistic isolate. It shares no structural, grammatical, or etymological relationship with any other language on Earth. Not with Spanish, not with French, not with the Indo-European language family that covers virtually all of Europe and much of Asia. Euskera is alone in the world, an island of sound, a grammar that belongs to no family, a vocabulary that arrived from somewhere no linguist can trace. And the Basque people have the highest concentration of Rh-negative blood on the planet. This coincidence, the linguistic isolation and the blood type anomaly appearing together in the same ancient mountain people, is the kind of detail that, once you notice it, refuses to leave the mind quietly. Rest in that thought for a moment. Let it be slow and soft. You don't need to solve it tonight. Nobody has solved it in 87 years of trying. Just let it breathe beside you in the dark, like an old question that has learned to be patient. In 1940, just 3 years after their initial discovery, Landsteiner and Wiener would publish their findings fully. The same year, a doctor named Philip Levine would make a separate, devastating discovery about what this blood factor means for pregnancy. And that discovery, that is the next part of our long journey. The place where the science becomes personal, intimate, deeply, achingly human. But for now, just hold this. A protein, an absence, a mystery born in a quiet laboratory. And already, even in these first years of discovery, a pattern in the data that doesn't quite fit the theories we build to explain it. Let your eyes grow heavy now. Let the numbers and the names drift like mist. You're safe here. The story continues whether you're fully awake or gently drifting. These words will find you wherever you are. Breathe in. Hold it gently for just a moment. And breathe out. We're in a tender place now. A place that requires the softest kind of attention. The kind you might offer to something fragile that you are carrying carefully across a room. This part of the story is about mothers. About the extraordinary, incomprehensible biological commitment of pregnancy, and about a particular cruelty that the Rh factor inserts into that commitment, quietly, invisibly, without malice, without intention, but with devastating consequence. It is 1940. Philip Levine is a young physician in Newark, New Jersey. He is studying a tragedy that has been recurring with heartbreaking frequency in obstetric wards across America and Europe for as long as records have been kept. Babies born severely ill, jaundiced, their skin and eyes stained yellow by the overflow of bilirubin from destroyed red blood cells. Profoundly anemic, sometimes stillborn, sometimes alive but fading, their small hearts working furiously to compensate for blood that is being attacked from within. This condition has had many names over the centuries. The Romans described it. Medieval midwives documented it. It appeared in the records of every hospital that carefully noted its sad outcomes. But no one understood why. Levine, working with the newly identified Rh factor, begins to see the pattern. The mothers of these sick babies are almost always Rh negative. The fathers, in the cases he can examine, are typically Rh positive. And the babies, inheriting Rh positive blood from their fathers, are born into a body whose environment, their mother's blood, has without any cruelty of intent, learned to see them as enemy. Let me describe the mechanism slowly, because it is important, not so you can memorize it, but so you can feel it. Feel the strange, terrible intimacy of it. During pregnancy, the placenta maintains a careful separation between maternal and fetal blood. These are two separate circulatory systems, running in extraordinary proximity, but designed not to mix. The placenta is the bridge, the magnificent, temporary organ that transfers oxygen and nutrients from mother to child without their bloods needing to merge. But during pregnancy, particularly during labor or delivery, small amounts of fetal blood can cross into the maternal circulation. Microscopic breaches, tiny leaks at the placental border. If the baby is Rh positive and the mother is Rh negative, something happens in the mother's immune system that is, from a purely biological standpoint, entirely reasonable. Logical. Her immune system is doing exactly what immune systems evolved to do. It encounters a foreign protein, the Rhd antigen, and it responds. It builds antibodies, anti-D antibodies, specialized, targeted, persistent. The mother's immune system is thorough and careful and precise. It creates a memory, an immunological record of this foreign invader, and it stands prepared. The terrible timing of this mechanism is that the first pregnancy often escapes the worst consequences. The immune response builds slowly. The antibodies that are created are of a type, IgM, that are too large to cross the placenta. The first baby, though the sensitization is occurring in the background, is often born safely. But the second pregnancy, the third, if these subsequent babies are also Rh positive, the mother's immune system, now primed, now trained, now holding its anti-D antibodies ready, responds faster and with catastrophic efficiency. This time, smaller IgG antibodies are produced. And IgG antibodies can cross the placenta. They cross into the baby's blood. And there, in the warm, closed world of the womb, they attack. They bind to the Rh positive red blood cells of the developing child and mark them for destruction. The baby's own circulatory system, the very foundation of its life, is systematically dismantled by antibodies that arrive from the person who loves it most completely. The person whose entire biological existence is, in every other way, organized around its protection and survival. This is hemolytic disease of the newborn, erythroblastosis fetalis, a condition with a name as complicated as the tragedy it describes. And here, right here, is where the evolutionary paradox sharpens into something genuinely bewildering. Evolution is ruthless about reproductive fitness. It eliminates, without sentiment or hesitation, traits that reduce a species' ability to successfully produce offspring. Rh negativity demonstrably reduces reproductive fitness in Rh negative women who partner with Rh positive men, which, given that Rh positive is overwhelmingly dominant, is most partnerships. In a population where this mattered, where Rh negative mothers were regularly losing second and third children, where family sizes were being reduced by this quiet biological conflict, Natural selection should have responded, should have gradually increased the frequency of Rh-positive genes, slowly diluting Rh-negative frequency across generations. In populations where Rh-negative was merely a neutral variation, having neither benefit nor cost, it might persist at some level, though typically drift would reduce it over time. But for it to persist at 15% globally, to cluster at 35% in isolated ancient populations, to have remained stable across thousands of years of human reproduction, that requires explanation. That requires something we haven't yet named. Some force, some advantage, some compensatory benefit, some evolutionary counterweight. That balances the reproductive cost. That makes Rh negativity worth keeping, worth preserving in the human genome across the vast, slow centuries of deep time. What could that be? What possible advantage could compensate for the cost of maternal-fetal blood conflict? That is the question that will occupy us for much of this long, slow night. But for now, for this quiet, tender scene, I want to leave the science aside for a moment and simply sit with the human reality of what we've described. Mothers who lost babies they did not understand. Across centuries, across millennia, the grief of it quiet and unexplained, attributed to God's will, to fate, to the particular cruelty of chance. Women who carried their lost children gently, not knowing that the answer was in their blood, not knowing that the same blood running in their veins, the same ancient, quiet mystery, was both the vehicle of their loss and the remnant of something very, very old. Let that sit softly. Let your breathing slow. You are safe. Whatever your blood type, wherever you are in the dark tonight, you are safe. The history is not yours to carry. It belongs to deep time, to the long record, to the slow and patient work of science and understanding. The grief of those mothers has a name now, has a treatment, has a small, extraordinary injection called Rh immunoglobulin, ROGAM, that has, since the 1960s, quietly saved hundreds of thousands of lives. That is worth knowing, that in the long arc of the mystery, the kindness comes, the understanding arrives, and the grief finds, eventually, its medicine. Sleep is beginning to pull at the edges of your awareness now, isn't it? Let it. You can drift toward it without losing the story. The story will still be here, moving quietly beneath your sleep like a river beneath ice, present, flowing, ancient, high in the mountains. There are mountains in the world that feel old in a way that goes beyond geology. Mountains whose shadows seem to carry memories, whose valleys hold silence the way a cupped hand holds water, carefully, completely, without losing a drop. The Pyrenees are such mountains. They rise between France and Spain in a long, dramatic spine. Limestone and granite, an ancient sedimentary rock laid down when this part of the world was an inland sea. They are not the highest mountains in Europe, but they have a quality of permanence, of settled permanence, that the higher, sharper peaks of the Alps somehow lack. The Pyrenees feel inhabited in a way that goes beyond the villages clinging to their slopes. They feel inhabited by time itself. And in the valleys of these mountains, on both the French and Spanish sides of the high ridge, live a people who are, by every measure that population genetics and linguistics can apply, among the most remarkable anomalies in the entire human story. The Basque people, their name for themselves is Euskaldunak, those who speak Euskera. And Euskera is the key, because Euskera is not like other languages. It is not simply different. Many languages are different from one another. It is structurally, fundamentally, categorically unlike every other language spoken by humans on Earth. Consider, most languages in Europe belong to the Indo-European family. French and Spanish and Italian and Portuguese are Romance languages. They descend from Latin. English and German and Dutch are Germanic. Russian and Polish and Czech are Slavic. Even the seemingly alien languages of Greece and Armenia and Farsi in Iran are Indo-European cousins. Their deepest grammatical bones are recognizable to a trained linguist as kin. Then there are the Finno-Ugric languages, Finnish, Estonian, Hungarian, which form their own separate family. But Euskera? Euskera stands alone. It has been spoken in the Pyrenean region for so long that it predates the arrival of the Indo-European languages entirely. Linguistic and genetic evidence now suggests that the Basque people, or their direct linguistic ancestors, have been in this region since before the Neolithic Revolution, before farming, before the great migration of agricultural peoples from Anatolia that reshaped the genetic landscape of Europe approximately 8,000 years ago. The Basque people, in a very real sense, are the remnant of the first modern Europeans, the hunter-gatherers of the Upper Paleolithic, the people who painted the horses and bison and hands on the walls of Altamira and Lascaux. Those deep, dark, torchlit caves where the earliest known art breathes in the underground dark. They are still there, adapted, changed, modern, of course, but genetically distinct, linguistically isolated. A thread of very great antiquity still running, somehow, into the present day. And they have the highest concentration of Rh-negative blood on Earth. Now let us sit with that slowly. The Basque people are the oldest continuous human population in Western Europe. Their language predates the arrival of farming. Their genetics show remarkable isolation, a reduced diversity suggesting a long period with limited outside mixing. And their Rh-negative frequency, 30 to 35% in the general population, higher in isolated communities, is more than double the global average. This is not coincidence in the statistical sense. When a trait clusters in a specific identifiable, genetically distinct population, and that population happens to be one of the oldest in the region, the clustering is telling us something. It is a signal, a pattern, a data point that points backward into time, like an ancient compass needle swinging toward magnetic north. What it seems to suggest, carefully, cautiously, in the way that good science always moves, is that Rh negativity is old, that it was present in the populations of pre-Neolithic Western Europe at high frequency, and that as those original populations were gradually mixed with, and in most places largely replaced by, incoming Neolithic farmers from Anatolia, the Rh-negative frequency was diluted, averaged downward, spread thinner across the gene pool of the new, mixed Europe, except in the Basques, who, tucked into their mountain valleys, protected by altitude and geography and cultural insularity, maintained a greater degree of their original genetic inheritance, including, it seems, whatever mechanism or evolutionary history led to the high frequency of Rh negativity in the first place. This does not, it should be noted, mean that Rh-negative blood is exclusively European or exclusively Basque. It appears in populations across the world, though at lower frequencies. It appears in North Africa, particularly among the Berber people of the Atlas Mountains, another ancient, isolated population with a long history in their particular geography. It appears in the Arabian Peninsula and in the Caucasus region, that ancient crossroads between Europe and Asia. But the pattern is consistent. It tends to cluster in populations with long histories of isolation, of reduced outside admixture, of genetic bottlenecks, the narrowing of a population to a smaller founding group, concentrating whatever traits that group carried. The Basque anomaly is not an exception to a rule. It is the clearest example of the rule itself, a lighthouse in the fog of deep time, pointing toward an origin we cannot yet fully see. Let the mountains settle into your mind now. Those old limestone peaks, those quiet valleys, the villages where Euskara is still spoken in the evenings, still alive, still grammatically impossible, still belonging to a family of one. Imagine you're sitting at the edge of one of those valleys as the sun sets behind a ridge. The air smells of pine resin and cold stone, and the particular mineral cleanness of mountain water. Below you, the lights of a village are beginning to come on. Warm yellow squares of light against the darkening slope. And somewhere in that village, in the blood of the people moving quietly through their evenings, is the same ancient frequency, the same proportion of absence and presence, the same molecular signature that connects them, in ways they may or may not know, to the Paleolithic hunters who painted the caves. You are safe here on this hillside. The night is coming gently. The story continues below, in the lamplight of the village. Rest. There is a particular kind of map that scientists make. It is not a map of roads or borders or mountain ranges, though those features might appear in the background as reference points. It is a map of something invisible, something carried, a map of human biology as it distributes itself across the surface of the Earth, following patterns that are part migration history, part random genetic drift, part the slow and inexorable pressure of natural selection working across geological spans of time. These maps are, to those who learn to read them, among the most beautiful and haunting objects in science. Tonight we are looking at one, a world map. Imagine it spreading out before you in the soft dark. Not the hard plastic of a classroom globe, but something older. Something like a map drawn on stretched animal skin with pigments made from ochre and charcoal and the crushed shells of river mollusks. Soft at the edges, rich with color. And across this map, we are painting in the frequency of Rh-negative blood. Where it is high, the color is deep, the particular rich amber of old firelight. Where it is low, the color fades to the palest possible trace. Where it is effectively absent, the skin of the map shows through uncolored. Watch what emerges. Western Europe glows. Not uniformly. The signal is strongest in the Pyrenean region, that Basque mountain territory we have already visited. But it extends through the Iberian Peninsula, through France, through the British Isles, particularly Ireland and Scotland and Wales, those Celtic peripheries where ancient pre-Indo-European genetic signatures are also, not coincidentally, strongest. The British Isles, where Rh-negative frequency runs between 15 and 17%, where the people of the western coasts, the Welsh, the Irish, the Scottish Highlanders, carry frequencies higher than their eastern neighbors, where archaeology keeps finding evidence of populations that predate the Bronze Age migrations and maintain a degree of genetic continuity with the original post-Ice Age settlers of the islands. Moving south across the map, North Africa brightens. The Atlas Mountains of Morocco, Algeria, and Tunisia, home to the Berber people, the Amazigh, whose name means simply free people, and whose culture and language have endured, with remarkable persistence, for thousands of years despite the successive waves of Phoenician, Roman, Arab, and Ottoman influence that have passed through North Africa like tides. Among the Berber, Rh-negative frequencies reach 29% in some populations, a remarkable concentration in the world's second-largest continent, which overall has very low Rh-negative frequency. The Berber, like the Basque, are a people with a long history of geographic and cultural isolation. The Atlas Mountains are not the Pyrenees, but they serve a similar function, a refuge of high terrain where ancient populations could persist with reduced outside admixture. And like the Basque, the Berber languages are a linguistic puzzle, Afroasiatic in family classification, yes, but so old and so internally diverse that some linguists have suggested the Berber languages might represent the earliest split within the Afroasiatic family, a divergence of extraordinary antiquity. Moving east across the map, the Caucasus region brightens slightly. Georgia, Armenia, Azerbaijan, that ancient mountain corridor between the Black Sea and the Caspian, where human populations have been mixing and stratifying for tens of thousands of years. Rh-negative frequencies here hover between 10 and 15%, below the Western European peak, but above the Asian average. The Arabian Peninsula carries a moderate signal. Some populations in Yemen and Saudi Arabia showing frequencies between 8 and 12%. And then, as we move further east into South Asia, into East Asia, into the vast populations of China and Japan and Southeast Asia, the color fades almost completely. Rh-negative blood among Han Chinese populations is approximately 1%. In Japan, roughly 0.5%. In many indigenous populations of the Americas, Sub-Saharan Africa, and the Pacific Islands, it approaches 0. Now step back. Look at the whole map at once. What do you see? Not a random scatter. Not an even distribution that might suggest a mutation arising independently in many places, but a gradient, a pattern, a concentration in the west and northwest of Eurasia, in isolated mountain populations, in people with long histories of geographic and cultural insularity, and a near absence everywhere else. This pattern tells a story about where Rh negativity originated, and how, or rather, how slowly it spread, or did not spread. Because here is the crucial detail that the map is quietly insisting upon. The distribution of Rh negativity does not look like a trait that conferred a strong, universal advantage. If it did, if being Rh negative was highly beneficial in some way, it would have spread. Natural selection would have carried it outward from its origin point, increasing its frequency gradually in population after population. We would see a different map, a more homogeneous map. Instead, the map shows us a trait that is staying where it is, concentrated in its original location, diluted but not eliminated in the populations that absorbed some of the original carriers, nearly absent in populations that never encountered those original carriers, or encountered them only minimally. This is the behavior of a trait with a complicated history, one that might have been advantageous in a specific environment or context, and neutral or slightly costly outside that context. One that has been held at a particular frequency not by strong selection in either direction, but by some combination of founder effects, genetic drift, and perhaps a very specific compensating advantage that we haven't fully identified yet. The map is a mystery made visible, a frozen moment in the story of a long migration, a long dispersal, a long and slow dilution. Let your eyes drift closed now. Imagine the map softening, the colors blurring like watercolors in rain, the borders dissolving, until what remains is not a political or genetic map at all, but simply the Earth, turning slowly in the dark, holding its human cargo gently, carrying its mysteries across the centuries with the patient indifference of deep time. You're somewhere on that map, a single warm point of light in the pattern, connected to the pattern, part of the pattern. Sleep, if you're ready. The map will be here when you wake. The mystery will still be unresolved. That is what makes it beautiful. Let us sit very quietly with a contradiction, a genuine one, not the manufactured paradoxes of philosophical thought experiments, or the theatrical contradictions of debates designed to produce heat rather than light. A real, hard, data-supported contradiction, the kind that makes careful scientists slow down and stare at their data the way you might stare at a word you've written so many times it has temporarily lost its meaning. Evolution does not preserve what costs. This is not quite a law in the strict sense. There are edge cases, complications, interactions between traits that can preserve costly ones indirectly. But as a general operating principle of natural selection, the mechanism that has been shaping life on Earth for approximately 3.5 billion years, it holds up with remarkable consistency. Traits that reduce reproductive success are gradually eliminated. Traits that increase reproductive success are gradually spread. Traits that are neutral drift. They wander randomly, their frequencies shifting like sand dunes in an inconsistent wind, tending neither upward nor downward with any strong directionality. Rh negativity. In Rh-negative women, carrying Rh-positive babies reduces reproductive success. Not always. Not in every pregnancy. Not catastrophically in every case. But measurably, quantifiably, real. Before modern medicine, before Rh immunoglobulin, before the understanding of sensitization, before blood typing was routine, Rh-negative women in populations with high Rh-positive frequency would have been losing second and third children at elevated rates. Their reproductive output would have been statistically lower than their Rh-positive counterparts. Over thousands of generations, over the tens of thousands of years that modern humans have been living in organized social groups, this should have mattered. The mathematics of population genetics is patient. It doesn't need large effects. A small reproductive disadvantage, applied consistently across thousands of generations, is enough. Slowly, glacially slowly, in human experiential terms, but very quickly in evolutionary terms, it should have reduced Rh-negative frequency, pushed it toward the edges, made it rare, and yet, 15%, globally, with higher concentrations in specific ancient populations. Let us do the thought experiment carefully. Imagine a founding population of, say, early modern humans arriving in Western Europe somewhere between 40 and 50,000 years ago. Among this founding population, some percentage carries Rh-negative blood. Let's say 15%, close to the modern global average, or perhaps higher, if the founding group happened to carry it at elevated frequency. For the next 10,000 years, before farming, before large mixed populations, before the regular encounters with other large human groups that would change the genetic landscape, these people live in relatively small bands. Hunter-gatherers moving across a cold, rich landscape of mammoth grasslands and birch forests and the long fjords left by retreating glaciers. In small bands, the dynamics are different. if Rh-negative women are predominantly partnering with Rh-negative men, which, in a population where a third or more people are Rh-negative, is statistically likely, then the hemolytic disease problem largely disappears. Two Rh-negative parents produce Rh-negative children. No maternal-fetal conflict. No antibody attack. No lost babies. The cost of Rh negativity is, under these conditions, dramatically reduced. It might even, in certain environments, be close to neutral. And neutral traits in small populations do something interesting. They drift. They wander. In small founding populations, the genetics term is founder effect. A trait's frequency can increase dramatically by random chance, with no selection pressure required, simply by the accident of who survives, who reproduces, who carries the founding genetics forward. This might explain the vast concentration of founding population, perhaps the original post-Ice Age settlers of the Pyrenean region, who survived the last glacial maximum by taking refuge in the warmer southern pockets of Europe, carrying Rh negativity at elevated frequency, then remaining isolated enough that the frequency was preserved and perhaps even increased through drift. But this explanation, satisfying as it is in some respects, has limits, because 15% globally is not just a vast founder effect spreading outward. The distribution is too widespread, too consistent. It appears in populations with no genetic connection to Western Europe, those North African Berber populations, certain Arabian groups, Caucasian populations, in proportions that suggest either multiple independent origins or a very ancient origin predating the dispersal of modern humans from Africa. And here we encounter the deepest layer of the paradox. If Rh negativity is very ancient, if it arose in a common ancestor of all or most non-African populations, perhaps during or shortly after the migration out of Africa 70,000 years ago, then we need to explain not just why it has persisted, but why it appears at lower frequencies in African populations than outside Africa. This is a reversal of the usual pattern of human genetic diversity, which tends to be highest in Africa, the continent where our species has lived longest, and lower outside it, due to the bottleneck effect of the out-of-Africa migration. Rh negativity breaks this pattern. It is lower in Africa, higher outside, concentrated in specific ancient lineages that may represent some of the earliest non-African populations. What does that mean? Is it a mutation that arose in the out-of-Africa population itself, after the departure from the continent, but before the further dispersal into Europe and Asia? Is it older, carried by certain African populations that were among the primary contributors to the founding out-of-Africa group? Or, and here the science gets genuinely uncertain, genuinely contested, and genuinely thrilling, is there something about the environments encountered outside Africa that made Rh negativity not just neutral, but advantageous? Something that counterbalanced the reproductive cost with a survival benefit we haven't yet fully characterized? We are going to explore the proposed answers to these questions as our night continues. But for now, for this scene, in this quiet moment, I want you to simply appreciate the depth of the puzzle. This is not a trivial mystery. It is not a gap in our knowledge that simply hasn't been filled yet by obvious available data. It is a genuine, deep, unresolved scientific question, sitting in plain sight, written in the blood of 1 in every 7 humans alive today, being carried by those humans as they sleep and wake and love and work and build their quiet lives under the same stars that shone over Paleolithic Europe. The paradox is real. The silence is real. The question is real. And you're carrying part of the answer in your veins, whether you know your blood type or not. There's a theory, a serious one, peer-reviewed, published, debated in academic journals with footnotes and methodology sections and careful statistical analysis. Not a fringe idea or an internet speculation, but a genuine scientific hypothesis proposed by researchers who have spent careers thinking about the intersection of infectious disease and human genetics. The theory goes like this. Perhaps Rh negativity is not a cost at all in the broadest evolutionary context. Or more precisely, perhaps it is a cost in one specific area, the pregnancy complication, that is balanced by a benefit in another area. A benefit large enough in the environments where our ancestors lived to counterbalance the reproductive losses. And the proposed benefit involves one of the oldest and most intimate relationships in the human story. not a relationship between people. A relationship between people and parasites. Specifically, Toxoplasma gondii. Let the name sit for a moment. Toxoplasma gondii. It sounds ancient. Because it is ancient. One of the most successful parasites in the history of life on Earth. A single-celled organism that has, with extraordinary evolutionary sophistication, learned to complete its life cycle across multiple host species, manipulating host behavior in ways that seem almost impossibly clever for something without a nervous system. Toxoplasma is a protozoan parasite. Its definitive host, the host in which it reproduces sexually, is the cat. Domestic cats, wild cats, lions, cheetahs, any member of the Felidae family. In the cat's intestine, Toxoplasma produces oocysts, and those oocysts are shed in the cat's feces. From there, the parasite's remarkable journey continues. Rodents, mice, rats, ingest the oocysts. The parasite insists in their brain tissue. And here is the part that has fascinated and disturbed neuroscientists for decades. It appears to alter the rodent's behavior. Mice infected with Toxoplasma lose their innate fear of cat urine. Some studies suggest they are actually attracted to it. In their compromised, manipulated state, they approach rather than flee from cats. They are, as a consequence, significantly more likely to be eaten, which is, of course, exactly what Toxoplasma needs. Because inside the cat's gut, the parasite can complete its sexual reproductive cycle. Toxoplasma manipulates the brain of its intermediate host to engineer its own transmission. In humans, who can be infected through contact with cat feces, through undercooked meat, through contaminated water, Toxoplasma also insists in the brain. The infection is called toxoplasmosis. In healthy adults with intact immune systems, it is typically asymptomatic. Most people who carry it, and globally, estimates suggest between 30 and 50% of humans are infected, don't know it. But the effects are subtle and real. Studies have found correlations between Toxoplasma infection and slight changes in risk-taking behavior, reaction times, and personality traits in humans. The parasite may be exerting its behavioral manipulation in us, too, though the effects are far more diffuse and contested than in rodents. And crucially, during pregnancy, if a woman is newly infected with Toxoplasma while carrying a child, the parasite can cross the placenta and cause severe damage to the developing fetus. Congenital toxoplasmosis, hydrocephalus, eye damage, brain damage, stillbirth. Now, the hypothesis. Researchers Flagler and colleagues have proposed that the Rh blood group system and Toxoplasma may have a relationship. Specifically, that Rh-negative blood may confer some resistance to Toxoplasma infection or to the more severe effects of the parasite. The mechanism is not fully elucidated. This is not a proven, established biochemical pathway, but a hypothesis based on population-level observations. But the proposal is that the Rhd protein, or its absence, may affect the membrane properties of cells in ways that interact with the parasite's ability to infect them. If Rh-negative individuals are less susceptible to Toxoplasma infection, or to the behavioral manipulation it induces, or to the severe fetal consequences during pregnancy, then the evolutionary math changes dramatically. In an environment where Toxoplasma was a significant source of fetal loss and infant mortality, Rh-negative mothers might have actually had a compensatory advantage in their pregnancies. The very pregnancies where hemolytic disease threatened their Rh-positive babies might have been, on a population level, healthier with respect to toxoplasmosis. A cost balanced by a benefit. A trade-off written in the molecular language of blood proteins and immune responses and the long coevolution of hosts and parasites. Now, and this is important, this hypothesis is not proven. It is contested. Other researchers have found conflicting data or proposed methodological concerns about the studies supporting it. The relationship between Rh blood group and Toxoplasma susceptibility is real enough to study, but not settled enough to call established science. But it points towards something real, the idea that Rh negativity's persistence might be explicable through host pathogen coevolution, through the arms race between human immune systems and the organisms that have been trying to exploit them for millions of years. This is a well-established mechanism for maintaining genetic diversity. Sickle cell anemia, a devastating disease in its homozygous form, persists in African populations because the heterozygous form, sickle cell trait, provides significant resistance to Plasmodium falciparum malaria. The cost and the benefit exist side by side in the same genetic variant, maintained in the population because the environment creates selection for both. Could Rh negativity be another such trade-off? Could the cost, the maternal-fetal conflict, be balanced by a resistance to something ancient and ubiquitous and harmful? We don't know yet. And that uncertainty, that honest, careful, scientific uncertainty, is itself beautiful. It means the question is still alive, still being turned over by careful minds, still being tested against new data. The parasite hypothesis is one lamp in the dark, not the only lamp, not, perhaps, the brightest, but a real light, casting its particular illumination on a very old mystery. Let that light be soft now as you breathe. The mystery is patient. It has been patient for hundreds of thousands of years. It can wait a little longer while you rest. We need to go further back, much further back, past the Basque mountain villages with their lamplight evenings and their ancient, impossible language, past the upper Paleolithic cave artists with their ochre-stained fingers and their fire-lit galleries, past even the out-of-Africa migration 70,000 years ago, that great departure from the continent of our origins. We need to go back to a time when modern Homo sapiens, our direct ancestors, shared this planet with beings who were not quite us, who were recognizably human in many ways, who used fire and made tools and buried their dead, and perhaps sang, perhaps dreamed, perhaps looked up at the stars with the same expanding sense of smallness and wonder that we experience when we look up tonight. The Neanderthals, Homo neanderthalensis, named for the Neander Valley in Germany, where their bones were first formally described by science in 1856. But they were in Europe and Western Asia for perhaps 300,000 years before that discovery. They were there when the first modern humans arrived, and they persisted in dwindling populations until approximately 40,000 years ago, when they disappear from the fossil record, their extinction coinciding roughly with the spread of modern Homo sapiens across the same territories. For most of the 20th century, the relationship between Neanderthals and modern humans was framed as one of displacement, replacement. Modern humans arrived, with their superior technologies and their symbolic thinking and their more complex social structures, and the Neanderthals, already stressed by a difficult Pleistocene climate, declined, were outcompete, fated. This narrative was clean, comfortable, in its way. It put Neanderthals at a safe genetic distance from us, a cousin species that died out without contributing to our lineage. Then came Svana Pabo and the ancient DNA revolution. In 2010, Pabo and his team at the Max Planck Institute for Evolutionary Anthropology published something that rewrote the human story in a single paper. Using DNA extracted from Neanderthal bones tens of thousands of years old, the DNA degraded and fragmentary, and requiring extraordinary technical sophistication to sequence. They determined that modern humans outside Africa carry between 1 and 4% Neanderthal DNA in their genomes. Not zero, not a footnote. 1 to 4%. The clean narrative of simple replacement was wrong. Or rather, incomplete. Modern humans and Neanderthals interbred. Not extensively, not as equals in a continuous population, but at contact zones, probably in the Middle East, where the earliest modern human migrations from Africa would have encountered Neanderthal populations. There were meetings. There was mingling. Children born of both lineages who survived and whose descendants carried Neanderthal DNA forward into the expanding modern human population. Now, here is where we return a blood to the Rh factor, to the mystery that has been threading through this entire night's journey, like a quiet river running beneath the surface of everything. Neanderthals, as best as ancient DNA analysis can determine, were Rh positive. They carried the Rhd antigen. This is consistent with what we know about the Rhd gene and its deep evolutionary history. The Rh blood group system is ancient, predating the split between modern humans and Neanderthals by millions of years. Non-human primates are Rh positive. Ancestral hominin populations were almost certainly Rh positive. So, where did Rh negativity come from? If our common ancestor was Rh positive, and Neanderthals were Rh positive, and the vast majority of modern humans are Rh positive, then the absence of the Rhd antigen in 15% of modern humans is an event, a change, a deletion or deactivation of the Rhd gene that happened at some specific point in human evolutionary history. When? The genetic evidence is not yet precise enough to give us a confident date. But the distribution pattern, concentrated in Western Eurasian populations, particularly ancient isolated ones, is consistent with an event that occurred either before or during the early modern human occupation of Europe and Western Asia. Some researchers have proposed a tantalizing, deeply speculative, but not entirely dismissible possibility. What if Rh negativity arose not in modern Homo sapiens at all, but in a different hominin lineage, possibly an archaic human population whose genetic contribution to modern humans is yet to be fully characterized? We know now, thanks to Pabo's work and the subsequent explosion of ancient DNA research, that modern humans outside Africa carry contributions from at least two archaic populations, Neanderthals and the Denisovans, a group known almost entirely from a finger bone and a few teeth found in a Siberian cave, whose DNA tells us they were a distinct hominin lineage that also interbred with our ancestors. What if there is a third contributor, a ghost population, an archaic lineage that left its mark in modern human genetics in ways we are still learning to read? This is not established science. It is scientific speculation, the kind that serious researchers entertain cautiously as a possibility to be tested rather than a conclusion to be announced. But it is worth holding gently. The idea that the Rh negative blood type, this quiet, ancient, persisting mystery, might be a molecule-sized remnant of a meeting, an encounter in the deep past, before history, before language as we know it, before the first cave paintings were laid down on limestone walls. A gift, perhaps, from a lineage we have not yet fully named. A biological legacy of a meeting between peoples who were very old and peoples who were very new, in a world that was very cold and very dark and very full of stars. Let that thought carry you for a moment. The stars are the same stars. The dark is the same dark. And in your blood, tonight, is a story that may reach back further than any story we have learned to tell. Let us slow down even further now. We have been moving through deep time, through Neanderthal meetings and Basque mountains and Paleolithic migrations and parasitological hypotheses. Let us come back, gently, to the body, to the immediate, intimate, warm reality of blood moving through living tissue. Because one of the things that makes the Rh story scientifically compelling, and not merely historically interesting, is the question of whether Rh negative blood behaves differently in the body in ways beyond the pregnancy complication. And the answer, gathered carefully from decades of immunological research, is possibly yes, in ways that are subtle, cumulative, and deeply interesting. The immune system is the body's most complex information processing network, more complex in many ways than the brain, though the brain, naturally, tends to take credit for most of the interesting work. The immune system must distinguish, continuously and with extraordinary precision, between self and non-self, between the molecules that belong to the body and the molecules that don't, between the proteins produced by your own cells and the proteins introduced by bacteria, viruses, parasites, and allergens. This distinction, self versus non-self, is the fundamental operating principle of immunity. And blood type antigens are, in the language of the immune system, identity markers, molecular flags. The ABO blood group antigens are on the surface of red blood cells and many other cell types, and they are among the strongest self-markers in human biology. This is why blood type compatibility is so critical in transfusions. The wrong blood type triggers an immediate, catastrophic immune response because the immune system recognizes those foreign antigens as decisively, dangerously non-self. The Rhd antigen is, by comparison, a less aggressive trigger, but it is a trigger. When Rh-negative individuals receive Rh-positive blood, whether through transfusion or through the pregnancy mechanism we discussed, they could develop anti-D antibodies. These antibodies mark the Rh-positive blood cells as foreign and target them for destruction. What researchers have been exploring in recent decades is whether the presence or absence of the RHD antigen affects the immune system's overall calibration in ways that extend beyond this specific antibody response. Some findings are intriguing. Studies have found potential correlations between Rh blood group and susceptibility to certain infections. Some data suggests Rh-negative individuals may respond differently to certain viral infections, though the mechanisms and the consistency of these findings remain contested and incompletely understood. There is also the question of the RHD antigen's role in the cell membrane itself. The Rh proteins are not simply identity flags. They are structural components of the red blood cell membrane, embedded in the lipid bilayer and involved in its organization and function. In Rh-null individuals, those very rare people who express none of the Rh proteins at all, a condition so rare it has been found in fewer than 50 people worldwide, the red blood cells are structurally abnormal. They are stomatocytic, meaning they have a cup-shaped deformity rather than the normal biconcave disc shape. They have a shortened lifespan. They are fragile. This suggests that the Rh proteins are doing something structural, something important for the integrity of the red blood cell itself. What does the complete absence of RHD, specifically the Rh-negative condition, mean for cell membrane structure and function? The answer is not fully worked out. The cells appear normal by standard measures. They function normally in most observable ways. But the membrane is, at the molecular level, organized slightly differently in the absence of the RHD protein. Whether this difference has functional consequences, whether it changes how pathogens interact with the cell, whether it affects the cell's behavior in inflammatory environments, is an active area of inquiry. There is also an intriguing observation in the domain of allergy and immune reactivity. Some population-level data suggests that Rh-negative individuals may have somewhat different patterns of immune reactivity compared to Rh-positive individuals. Differences in rates of certain autoimmune conditions, in allergy profiles, and inflammatory responses. These associations are statistical and observational, not mechanistically explained, and must be interpreted cautiously. But they add to a picture that is slowly, carefully being assembled, that the Rh blood group system is not simply a label, not simply a transfusion compatibility marker and a pregnancy concern, but a deeper immunological characteristic, one with broad, if subtle, effects on how the immune system processes the world. And if the immune system is the body's way of engaging with its environment, its biological world of pathogens and allergens and cellular debris, then Rh blood group may, in ways we are still learning to describe, shape the nature of that engagement. The body, it turns out, is a very old laboratory. It has been running experiments in the form of immune responses, genetic variations, evolutionary pressures, for longer than we have been watching. And the Rh factor is one of those experiments, still in progress, still producing data that we are only beginning to know how to read. Take a slow breath now. Feel your immune system running quietly in the background. That enormous, silent, tireless operation of surveillance and response and memory and precision. Feel it doing its work without requiring your attention, without asking for your conscious participation. It is ancient. It is yours. It is, in its current form, the result of millions of years of refinement and adaptation and the particular history of the people who carried your genes forward to this moment. It is, in the most literal sense, your inheritance. Rest in that. We have lingered in the mountains of the Basques. Let's travel now, slowly, the way old maps suggest travel, with the awareness that the journey itself contains as much meaning as the destination, with the understanding that every place we visit is a chapter in a story that doesn't have a beginning we can reach or an ending we can see. We move east and south, to the Atlas Mountains. The Atlas Range extends in a broad arc across Morocco, Algeria, and Tunisia, the northwest shoulder of Africa, the region the ancient Romans called Mauritania, and which has been, for the entirety of recorded human history and long before, home to the Berber people, the Amazigh. Sit with this landscape for a moment. The high Atlas of Morocco in winter, peaks above 3,000 meters, draped in snow that catches the low sun and holds it, turning the morning sky a particular shade of blue that painters try and rarely capture. Below the snowline, cedar forests and juniper scrub. Below that, The pale ochre of the lower slopes where Berber villages of Pizay, sun-dried earth brick, stand as they have stood for centuries, their flat roofs arranged in terraces against the hillside. The Berber people have lived in this landscape, or landscapes very like it, for a very long time. Archaeological evidence suggests human habitation of North Africa extending back hundreds of thousands of years. The Capsian culture, a Mesolithic people of northern Africa, was here 10,000 years ago. Before them, people whose genetic contribution to modern North Africans can still be detected, ancient enough to predate the Saharan Green Period, when the Sahara was a wet, inhabited grassland in North Africa, was connected to sub-Saharan Africa by passable terrain. The Berber languages are ancient. Their script, Tifinagh, is among the oldest continuously used writing systems in the world. Their cultural distinctiveness has persisted through Phoenician trade contact, through Roman colonization, through the Arab conquest of the 7th century CE, through Ottoman administration, through French and Spanish colonial rule. Layers of history pressing down, and the Berber identity persisting beneath each one, battered but unextinguished. An Rh-negative frequency among the Berber, in some populations, approaching 29%. Not Basque levels, but dramatically elevated above the global average. Elevated enough to be statistically significant. Elevated enough to add another data point to the pattern we have been tracing all night. Let us continue traveling to the Caucasus. That ancient region, the mountain spine between the Black and Caspian Seas, home to Georgia, Armenia, Azerbaijan, and the extraordinary linguistic diversity of Dagestan, where dozens of mutually unintelligible languages are spoken within a relatively small geographic area, has long been recognized as a place of deep human antiquity. Genetic studies confirm that the Caucasus region was one of the refugia, the warm pockets where human populations survived the worst of the last glacial maximum, a place where Ice Age people sheltered, maintained their genetic lineages, and then, as the climate warmed, expanded back out into Europe and Western Asia, and contributed their genetics to the populations that now inhabit those regions. Georgian, Armenian, and certain other Caucasian populations show Rh-negative frequencies in the 10 to 15% range, below the Basque Peak, but consistent with the broader pattern of elevated frequency in ancient, isolated, Western Eurasian populations. And now, because the pattern is global and the story is global, let's cast our gaze to the Americas. The indigenous peoples of the Americas, whose ancestors crossed from northeastern Asia into Alaska during the last Ice Age, when the Bering Land Bridge connected the continents, show a striking characteristic: extremely low Rh-negative frequency. In many pure indigenous American populations, Rh negativity approaches zero. It is effectively absent. This is entirely consistent with the pattern. If Rh negativity arose in Western Eurasian populations, in or near Europe and Western Asia, after the ancestral Native American population departed Asia approximately 15 to 20,000 years ago, then those founding Americans would not have carried it. They departed from a population that was itself largely Rh positive before the Rh negative variant, if it arose or increased in frequency later, had spread into the Eastern Eurasian population. The Native American genetic isolation from Western Eurasia preserved their original absence of Rh-negative blood, a molecular timestamp of when their ancestors' journey began. Similarly, in East Asian populations, Chinese, Japanese, Korean, Southeast Asian, Rh-negative frequency is typically below 1%. These populations' ancestors left Africa in the Great Out-of-Africa Dispersal and moved rapidly eastward, reaching Australia as early as 65,000 years ago and East Asia not long after. They carry, correspondingly, a genetic profile that suggests limited admixture with the Western Eurasian populations where Rh negativity is concentrated. And in sub-Saharan Africa, where human genetic diversity is greatest, where our species has its deepest roots, where populations have the longest uninterrupted evolutionary history, Rh negativity is also low, typically 2 to 3%. Present, but at a frequency consistent with occasional admixture with North African or Middle Eastern populations rather than an ancient indigenous origin. The map, traced out in the slow travel of this scene, is telling a consistent story. Rh negativity is a Western Eurasian signal. It points to a population, perhaps the descendants of those early modern humans who first occupied Europe and Western Asia, perhaps influenced by archaic human lineages we haven't fully characterized, in whom this blood characteristic became elevated and was subsequently maintained. It is the molecular geography of an ancient people. Their descendants are still among us, in the Basque Mountains, in the Atlas Range, in the Caucasian valleys, in the Celtic peripheries of the British Isles, and scattered, diluted but present, through the vast populations that descended from and mixed with those original carriers. You may be one of them, even if you don't know your blood type, even if you've never given it a thought, even if the idea is entirely new to you, arriving like a slow tide at the edge of your sleeping attention tonight. The pattern is real. The story is yours. Rest now in the warmth of that belonging. Let the science rest for a moment. Let the chromosome maps and the population genetics and the molecular biology settle into the background like the sound of a river that you've been walking beside for a long time. Still present, still real, still moving, but no longer demanding the front of your attention. Because humans have always had ways of explaining what they could not yet measure. Long before Landsteiner and Wiener were peering at blood samples in their New York laboratory, long before the electron microscope revealed the landscape of the cell surface, long before chromosomes were mapped and genes were sequenced, people noticed. They noticed difference. They noticed the people who seemed to run cold in hot weather and warm in cold weather, who had an unusual resistance to certain sicknesses, who seemed to feel the approach of storms before the clouds appeared, whose children, sometimes, were born ill in ways that broke hearts without explanation. Every culture that has been around long enough has built narratives around its biological outliers. The Celtic traditions of the British Isles are particularly rich in this regard. The concept of the seventh son of a seventh son, a lineage figure believed to carry healing powers, to have second sight, to exist at some intersection between the ordinary world and the unseen one. The idea of the changeling, the fairy child substituted for a human baby, explained in modern retrospect as perhaps an attempt to make sense of babies born with genetic differences that set them apart from their siblings. But it is in the folklore of blood, specifically, where some of the most haunting cultural echoes appear. Across multiple European traditions, particularly in the Celtic and pre-Christian Germanic worlds, there's a recurring motif of a lineage of people with unusual blood. Blood that is cold to the touch, or that heals differently, or that carries some particular spiritual quality that sets the bearer apart from ordinary humanity. The Irish concept of Sidhe, the fairy folk, the people of the mounds, describes beings who are neither fully human nor fully divine, who are old beyond ordinary reckoning, who are beautiful in an unsettling way, who sometimes merge with human lineages, producing descendants who carry unusual gifts. The Welsh Tylwyth Teg, the fair folk, serve a similar narrative function. They are the ancient people, the people who were here before, who retreated as the ordinary world expanded into the hills and the hollow mountains and the edge places between seen and unseen. The Norse Hulda folk, the hidden people, described in Icelandic folklore with such conviction that they have influenced Icelandic construction practices into the present day. Now, and let this land very gently because I'm not suggesting anything supernatural or literal, there's a fascinating cultural pattern here. Across multiple ancient European traditions, particularly in the regions where Rh-negative blood is most concentrated, there are persistent stories of a people who are old, who are different, who are connected to the land in ways that ordinary humans are not, and who sometimes enter into unions with ordinary humans, producing unusual descendants. This is not evidence. It is not science. It is folklore, the impressionistic, cumulative, emotionally coded knowledge of people who could feel and observe but not measure. But the overlap between the geographic distribution of these folklore traditions and the geographic distribution of elevated Rh-negative frequency is, at minimum, a poetic coincidence. At maximum, it might suggest something more human and less mystical. That in communities where some people were biologically different in detectable, subtle ways, different enough to sometimes produce complicated pregnancies, perhaps different in immune responses and physical characteristics. Those communities developed narratives to make sense of the difference, to explain it, to give it a name and a place in the cosmology. The people of unusual blood became, in story, the people of unusual origin. The biological mystery became a mythological mystery, and the myth carried the memory of the difference forward, in its own imprecise and emotionally resonant way, for thousands of years. There is also the more explicitly historical record. Medieval and early modern Europe had persistent legends about families, noble lineages in particular, who claimed ancestry from unusual, quasi-divine, or pre-Christian origins. The Merovingian dynasty of France, for example, those mysterious long-haired Frankish kings who ruled from the 5th through the 8th century CE, were surrounded by legends of unusual descent, of a founding ancestor who came from the sea, who was connected to older, stranger powers. The Merovingians, it is interesting to note, ruled a territory that overlapped significantly with the ancient Basque and pre-Celtic European regions of Western Europe. Again, not evidence. A pattern, a repeated motif, a story that keeps wanting to be told about certain lineages in certain places, a story about being old and different and carrying something in the blood that ordinary people do not. The folklore is the emotion of a fact that couldn't yet be measured. The science is the measurement of a fact that was always felt. Together, they tell a more complete story than either tells alone. Let the stories settle around you now like old tapestries on stone walls, faded by time, imprecise in their details, but warm, holding something real in their ancient imagery, something about the persistence of difference, about the long memory of blood, about the way certain truths find their way in a human consciousness, whether we have the tools to prove them or not. You are in good company in the dark tonight, surrounded by old stories and patient science, and the quiet warmth of a mystery that has been asking its questions for longer than civilization has existed. Rest in that company. Come back now to the laboratory. But a different laboratory than the 1937 New York room, where Landsteiner and Wiener first described the RH factor. A modern one, bright with the cold blue light of computational screens, filled with the quiet mechanical hum of next-generation sequencers. Machines that can read the entire 3 billion letter code of a human genome in a single day for a cost that would have seemed impossible just 20 years ago. This is where the RH story has moved in the decades since its initial discovery. From the serology laboratory, where blood typing was done with antibodies and visual reactions, to the genomics laboratory, where the story is read directly from the DNA. And what the DNA is saying is, in its quiet molecular way, extraordinary. The RH blood group system is encoded on chromosome 1, the largest human chromosome, in a region that contains two adjacent and highly similar genes, RHD and RHC. These genes are so similar to each other, sharing approximately 96% of their sequence, that they are believed to have arisen from an ancient duplication event, an ancestor gene that copied itself and then slowly diverged, producing two related but distinct proteins. This duplication is ancient. It predates the split between humans and other great apes. The RHD and RHC genes are found in chimpanzees and gorillas in recognizable form. The RH blood group system has been running in the great ape lineage for tens of millions of years. But here is where the human story diverges. In most humans, RH-positive humans, both the RHD and RHC genes are present and functional. The RHD gene is expressed on red blood cells, producing the RHD protein. The RHC gene is also expressed, producing the RHC protein complex. In RH-negative humans, specifically in the most common form of RH negativity found in Europeans, the RHD gene is simply absent, not mutated, not present in a broken form, gone, deleted from that region of chromosome 1. What occupies the space where the RHD gene should be is a hybrid sequence, the result of a recombination event where the flanking sequences of RHD fuse together, removing the gene entirely. This is called the RHD deletion, and it accounts for the majority of RH-negative phenotypes in people of European ancestry. This is important. A deletion is different from a point mutation or a regulatory change. A deletion is permanent, obvious, and requires a specific recombination event to create. It is the kind of change that happens once, or a small number of times, in evolutionary history, and then spreads through the population via inheritance. The Rhd deletion in European Rh-negative individuals is, as far as genetic analysis can determine, a single event, or a very small number of events. It arose in a specific individual, in a specific population, at a specific time, and was then passed down through the generations to the roughly 15% of modern Europeans and their descendants worldwide who carry it. When did this event occur? This is one of the most actively investigated questions in Rh genetics. The deletion must be old enough to have spread to the current frequency through normal inheritance and whatever selection pressures acted on it. Population genetic models, combined with estimates of how quickly neutral or slightly deleterious variants spread in populations of different sizes, suggest the deletion is likely at least 30 to 50,000 years old, placing it in the Paleolithic period, consistent with the distribution pattern we've been discussing. Some analyses suggest it could be significantly older. In non-European Rh-negative individuals, those from Africa, Asia, or the Middle East, the mechanism of Rh negativity is often different. Rather than the complete Rhd gene deletion seen in most Europeans, other populations carry point mutations within the Rhd gene that prevent the protein from being expressed on the cell surface. The gene is present but silent, broken rather than absent. This is potentially significant. It might mean that Rh negativity in non-European populations arose independently, separate events, separate mechanisms, producing the same functional phenotype through different molecular routes. If this is the case, then Rh negativity in, say, the Berber population might have a different genetic origin than Rh negativity in the Basque population, even though the blood type phenotype is identical. Or it might mean that both populations share a common ancestor in whom the Rhd change began, with different molecular consequences depending on which variant was inherited by which descendant population. The genomics of this are still being worked out. Ancient DNA analysis, the sequencing of DNA from archaeological human remains, will eventually provide clearer answers. When we can sequence the genome of Basque remains from 8,000 years ago, or Berber remains from the Mesolithic, or Caucasian remains from the Upper Paleolithic, and determine their Rh status directly, we will have a much clearer picture of when and where and how many times the Rh negative phenotype arose. That data is coming. It is being gathered now in laboratories across Europe and the Middle East by researchers who understand that the ancient DNA revolution is still, in many ways, just beginning. For tonight, for this slow, quiet journey through blood and time, what matters is this. Somewhere on chromosome 1, in your genome, is the answer to whether you're Rh positive or Rh negative. Whether the Rhd gene is present and functional, producing its protein on your red blood cells, or whether the space where that gene should be is empty, a deletion, an absence, a silence where a very old gene once was and now is not. That silence, if it is in you, is a very particular kind of inheritance. A remnant, a molecular fossil, the preserved record of a genetic event that happened to one of your ancestors in the deep and nameless past. And that ancestor's blood is, in the most literal sense, still running in you. Let your own blood move quietly now, its warmth familiar and comforting, carrying its ancient story through the geography of your sleeping body. There are, at this moment, fewer than 50 people in the world whose blood has a particular quality. Not 15% of the world's population. Fewer than 50 individuals, living their regular lives, cooking their dinners and reading their books, and walking through their ordinary days with extraordinary blood moving quietly through them. Their blood has been called golden blood, not metaphorically. That is the actual term used by some researchers in blood banks, golden blood, a name that arose partly from its extraordinary rarity and partly from the way it behaves in blood banking, as something of exceptional, irreplaceable value. The technical name for their blood type is Rh null. These individuals express none, zero, of the Rh proteins on their red blood cells, not just the Rhd antigen that distinguishes Rh positive from Rh negative, not just the Rhc proteins that define the C, E, and E antigens within the Rh system. All of them, gone, absent. the entire Rh protein complex, missing from the red blood cell surface. Rh null blood was first described in 1961 in an Aboriginal Australian woman. At the time, it was thought to be simply a curiosity, perhaps unique to her, perhaps an extremely rare genetic accident of no broader significance. But over the following decades, more Rh null individuals were found. In Japan, in Brazil, in Europe, in multiple different ethnic backgrounds, which suggested that the Rh null phenotype, while vanishingly rare, was not confined to a single population. Two distinct mechanisms were eventually found to produce Rh null blood. The first, called the regulator type, results from mutations in a gene called RHAG, which encodes a protein called Rh-associated glycoprotein. The RHAG protein is not itself an Rh antigen, but it is absolutely required for the Rh proteins to be expressed on the cell surface. Without functional RHAG, no Rh proteins appear, even if the RhD and RhC genes themselves are intact and normal. The genetic instructions are present. The proteins simply cannot find their way to the surface. The second, called the amorph type, results from mutations in both the RhD and RhC genes themselves, silencing the production of the Rh proteins directly. Both types produce the same phenotype, blood cells with no Rh antigens whatsoever. And here, the immunological consequences become almost impossibly challenging. Rh null blood is, in transfusion medicine, simultaneously the most universally compatible and the most universally incompatible blood type that exists. On one hand, because Rh null red blood cells lack all Rh antigens, they cannot trigger an immune reaction against any Rh antigen in any recipient. They are, in theory, the most universally transfusible red blood cells in the world. Anyone can receive Rh null blood without fear of Rh-mediated transfusion reaction. On the other hand, if an Rh null individual ever needs a blood transfusion, in an emergency, in surgery, after an accident, they can only safely receive blood from another Rh null donor. Because their immune system, having never encountered any Rh proteins, will mount a catastrophic response against red blood cells that carry them. Any transfusion of ordinary blood, Rh positive or Rh negative, could potentially trigger a severe, life-threatening hemolytic transfusion reaction, and Rh null donors are, globally, fewer than 50 people. The logistics of this are sobering. Rh null individuals who live with the knowledge of their blood type typically donate it regularly for storage and banking. There are networks connecting Rh null donors across continents, so that if one of them, or one of the small circle of people who might safely receive their blood, ever needs an emergency transfusion, compatible blood can potentially be sourced and transported. It is a quiet, remarkable system of solidarity built around an extreme biological rarity. But beyond the medical logistics, the Rh null phenomenon raises something philosophically interesting. These 50-odd individuals have the most primordially blank version of the Rh blood type. They lack what most humans have, not just partially, as Rh-negative people lack RhD, but completely. They are, in a specific molecular sense, outside the entire Rh blood group system. And their red blood cells are, as we noted in an earlier scene, slightly different. Stomatocytic, cup-shaped rather than biconcave, with a slightly shortened lifespan. This tells us something important about the Rh proteins in general. They are not vestigial decorations on the cell surface. They are functional components. Their absence has consequences, subtle ones in the case of Rh-negative, lacking only RhD, and more significant ones in the case of Rh null, lacking everything. The Rh proteins, in other words, are doing something important that we haven't completely characterized. They are part of the cell's architecture in a fundamental way, which makes their story, the story of how some humans came to lack one of them, and a tiny number came to lack all of them, even more interesting, even more worth the long, slow, careful attention of this night's journey. The 50 golden-blooded people alive today, somewhere on this earth, right now, in the same dark that holds all of us tonight. There are 50 people whose blood is unlike anyone else's on the planet, who carry, in its most extreme form, the absence at the heart of the Rh story. They do not know each other, necessarily. They live scattered across continents, in different cities, speaking different languages. But they are connected by the rarest molecular characteristic in human biology, a tiny constellation of extraordinary blood. A reminder that within the already remarkable mystery of Rh negativity, there are deeper layers still, depths within depths, mysteries nested inside mysteries, like a set of very old, very beautiful boxes, each one containing another. Sleep now if you can. The golden blood is out there in the dark, moving through 50 quiet hearts. And somewhere in the spectrum between ordinary blood and golden blood, you are. We have spent a long time in the darkness of the mystery, in the unresolved spaces, the paradoxes, the distribution patterns that defy simple explanation, the evolutionary mathematics that doesn't quite balance, the genetic archaeology that is still being assembled, piece by careful piece. Let's pause here, in this middle-later stage of our long night's journey, to acknowledge something important, something that arrived in the story in the middle of the 20th century, like a lamp being lit in a room where people had been sitting in the dark for a very long time. The lamp was small. It was a syringe, and what it contained, a preparation called Rh immunoglobulin, commercially known as Rogum, saved hundreds of thousands of lives in the decades following its introduction, and continues saving lives today. Let's walk slowly through how this came to be. By the late 1950s and early 1960s, the mechanism of hemolytic disease of the newborn was fully understood. Researchers had traced the entire pathway: the Rh-negative mother, the Rh-positive fetus, the sensitization event at delivery, the antibody formation, the subsequent pregnancies in which those antibodies crossed the placenta and attacked the fetal red blood cells. The question was, could it be interrupted? Could you prevent the sensitization from occurring in the first place? The answer, it turned out, was yes. And the logic behind the solution is one of those beautiful moments in the history of medicine where the insight, once articulated, seems almost obvious in retrospect, but required enormous creativity and careful experimentation to reach. The key insight was this: if you could clear the Rh-positive fetal red blood cells from the mother's circulation before her immune system had time to mount a full response and develop a lasting antibody memory, you could prevent sensitization entirely. And the way to clear them, counterintuitively, was to give the mother Rh antibodies. Not let her body produce them. Give them to her, from outside, as a prepared biological product. Administered shortly after delivery or after any sensitizing event, miscarriage, amniocentesis, trauma to the abdomen during pregnancy, these externally provided anti-D antibodies would bind to and facilitate the clearance of any Rh-positive fetal cells in the maternal circulation, removing them before the mother's own immune system could form lasting memories of them. It is, in essence, a kind of immunological interference, a preemption. You use the thing you're afraid of, antibodies against Rh-positive blood, to prevent the development of the thing you're afraid of. The clinical trials conducted in the early 1960s by teams including researchers like Vincent Frieda and William Pollack in New York and Ronald Finn in Liverpool demonstrated that this worked. Dramatically, the rate of Rh sensitization in Rh-negative mothers who received Rh immunoglobulin was reduced by approximately 99%, from a condition affecting approximately 1 in every 100 to 1 in every 250 live births, causing stillbirth, severe anemia, jaundice, kernicterus, death, to a condition that in treated populations essentially ceased to occur. Rogum was approved for clinical use in the United States in 1968. In the years and decades that followed, it was introduced across the developed world. Standard practice became the administration of Rh immunoglobulin to Rh-negative pregnant women at 28 weeks gestation and again within 72 hours of delivery. The condition that had been causing the death and disability of babies throughout all of recorded human history, that had taken children from Rh-negative mothers in ancient Rome and medieval England and 19th century America, without those mothers or their physicians ever understanding why, was, within a decade, effectively eradicated in populations with access to modern obstetric care. This is one of medicine's quieter triumphs, not as dramatic as the polio vaccine, not as celebrated as antibiotics or anesthesia, but in its effectiveness and its specificity, its targeting of a precise, understood mechanism. It is a perfect example of what science can do when it moves from mystery to understanding to intervention. The journey took about 30 years from Landsteiner and Wiener's 1937 discovery to the clinical introduction of Rogum in 1968. 30 years to travel from, "we have found a blood factor," to "we can prevent this from harming any more children." That is fast in the context of medical history. That is the pace of urgency meeting rigor. But there is a quiet complexity to this triumph that deserves acknowledgement. Rogum is made from human plasma. Specifically, from the plasma of Rh-negative individuals who have been deliberately sensitized to produce anti-D antibodies. Or, in more recent manufacturing approaches, from monoclonal antibody production. The original human plasma donors, the people whose antibodies formed the foundation of this life-saving medication, were, in the early years of its production, often Rh-negative prisoners in American correctional facilities who participated in sensitization programs. The ethics of this, using the vulnerability of incarcerated populations in the development of a treatment that would benefit others, is a complicated chapter in the history of medical research, one that deserves to be named and held honestly, even in a context like tonight's that is otherwise moving toward wonder and peace. Science's greatest gifts sometimes have complicated origins. That honesty is part of the full story. But the result, the life-saving result, is real and remarkable. Babies who would not be alive today are alive because of it. Families that would have been broken by unexplained loss were not broken. The molecular mystery that nature left unresolved in the human genome was met, not with a final answer about why it exists, but with a practical, compassionate, effective human response to the harm it can cause. That is, in its own way, a kind of miracle. Breathe slowly now. Feel the weight of all that saved life in the air around you. All those children and grandchildren of children who survived because science understood, because researchers worked, because human creativity met a biological problem with patience and rigor and care. You are surrounded by that history tonight. It is a good history to rest inside. We are living in a remarkable moment. A moment that would have seemed like science fiction to Landsteiner and Wiener. A moment that the researchers who first mapped the Rh blood group system, spending years doing painstaking serology, typing blood one sample at a time with antibodies and careful visual observation, could not have conceived of even in their most optimistic projections. In the last 15 years, the cost of sequencing a complete human genome has fallen from approximately $3 billion. The cost of the original Human Genome Project completed in 2003 to less than $1,000. And falling still. The trajectory of the technology is so steep that within a decade, whole genome sequencing at clinical quality may be available to anyone who wants it for the cost of a routine blood test. Meanwhile, direct-to-consumer genetic testing companies, 23andMe, AncestryDNA, MyHeritage, FamilyTreeDNA, have genotyped more than 40 million people worldwide. 40 million individuals who have sent a small vial of saliva through the mail and received, in return, a report on their ancestry composition, their genetic health markers, their potential carrier status for inherited conditions, and their connections to DNA relatives. Buried within those reports, for many customers, is their predicted Rh blood type. And what is emerging from this vast, crowdsourced pool of genetic data, supplemented by the ancient DNA revolution, which is now producing genomic data from thousands of archaeological human remains, is a picture of Rh-negative distribution through time that is beginning to test and refine the hypotheses we've been exploring throughout this night. The ancient DNA findings are particularly striking. Researchers at institutions including the Copenhagen Group, the Harvard Ancient DNA Lab, the Max Planck Institute, and many others have been systematically extracting and sequencing DNA from Mesolithic and Neolithic European human remains. And the results, when they are analyzed for Rh status, are beginning to tell a story. Western European hunter-gatherers, the people who inhabited Europe before the arrival of Neolithic farmers from Anatolia, approximately 8,000 years ago, show elevated frequencies of Rh-negative blood compared to the Anatolian farmers, who would subsequently largely replace them genetically across most of Europe. This is consistent with what the Bass pattern was suggesting all along. The Rh-negative signal in modern European populations may be, in large part, a remnant of the genetic inheritance of those original post-Ice Age European hunter-gatherers, the people whose descendants the Basque most directly represent, because the Basque region was among the places where that pre-Neolithic genetic inheritance was best preserved. The Neolithic farmers who swept into Europe from Anatolia, bringing with them wheat and barley and cattle and pottery and the agricultural way of life, were predominantly Rh positive. When they arrived and mixed with the hunter-gatherers, the result was a dilution of the Rh negative frequency across most of Europe. But in the refuges, the mountain margins, the western peripheries, the places where geography or culture or both maintained some degree of isolation from the incoming wave, the old signal persisted. This is a beautiful and clarifying story. It suggests that Rh negativity is not exotic or alien or unexplainable. It is simply old. It is the molecular fossil of an ancient European people, our collective Ice Age ancestors, whose genetic contribution has been diluted but not erased by the subsequent demographic transformations of European prehistory. The Basque people are not strange. They are ancient. The high Rh negative frequency in Celtic populations is not mysterious. It is ancestral. It is the preserved inheritance of people who were here in these cold green islands and these western mountains and these Atlantic shores, before farming, before metallurgy, before writing. And the 15% of the global population who carry Rh negative blood are, in this reading, the dispersed descendants of those ancient peoples. Their molecular legacy carried outward by migration and exploration and the slow mixing of populations across historical time. This does not solve every aspect of the evolutionary paradox. It does not explain why the trait was maintained at significant frequency rather than drifting downward under selection pressure. It does not identify the compensating advantage, if one exists, but it grounds the mystery in a human reality, in actual people, in an actual landscape, in an actual historical sequence that we can increasingly trace with remarkable precision. The ancestors whose blood may be running in your veins tonight, if you are Rh negative, were here during the Ice Age. They hunted aurochs in the birch forests. They fished the cold rivers of what would become France and Spain and Britain. They painted the walls of caves with images that are still, 40,000 years later, among the most moving and beautiful objects created by any human hand. They survived the last glacial maximum, the coldest period of the last Ice Age, when much of northern Europe was uninhabitable and the population of the entire continent might have numbered in the thousands. They survived in their southern refuges, the Iberian Peninsula, the Italian Boot, the Balkans. And when the climate warmed and the glaciers retreated, they expanded back into the reopening landscape. They were here. They persisted. And something of them, something molecular, something coded in the particular sequence of chromosome 1, persisted too, into the Mesolithic, into the Neolithic, into the Bronze and Iron Ages and the Roman period and the medieval centuries and the modern era, into you perhaps, into your sleeping body, into the blood moving quietly beneath the skin of your hand. That hand we noticed together at the very beginning of this journey. That connection, that thread of molecular continuity reaching from a Paleolithic hunter in a cold European landscape all the way to you here tonight, is not a metaphor. It is biology. It is real. And it is beautiful. We should talk about the other stories. The ones that exist at the edges of this mystery, in the darker corners of the internet, in the breathless voices of certain YouTube channels and podcasts and self-published books. The stories that have grown up around the Rh negative blood type, the way vines grow around old ruins. Opportunistically, prolifically, finding purchase in every crack and gap left by incomplete scientific knowledge. These stories are not science. I want to say that clearly, and then I want to say something equally important. They're not entirely without reason. Because a genuine mystery, a real, documented, scientifically unresolved question, creates a vacuum, and human minds, which are meaning-making engines of remarkable sophistication, will fill a vacuum with stories. It is not a flaw. It is the deepest characteristic of our species. The cave painters who covered the walls of Lascaux were filling the vacuum of unexplained experience with story. The myth-makers of every culture who built cosmologies to explain the thunder and the seasons were filling vacuums. Story is how we process mystery before science is ready to explain it. So the stories that have accumulated around Rh negative blood are, in their way, a deeply human response to a deeply human question. The most common ones go roughly like this. Rh-negative blood is of alien origin. Extraterrestrial visitors, at some point in prehistory, interbred with the human population and introduced this blood type. The Rh designation, the rhesus factor, is taken to mean that Rh-negative people lack the monkey blood, making them genetically distinct from the primate evolutionary line, genetically connected to something else. Or, Rh-negative blood is the blood of the Nephilim, the beings described in the book of Genesis as the offspring of sons of God and human women. The giant, powerful, divinely connected lineage that was supposedly wiped out by the flood. Or, Rh-negative people share ancestry with the inhabitants of Atlantis, with ancient advanced civilizations, with the bloodlines of the great mystical traditions. Or, Rh-negative individuals have unusual psychic abilities, higher rates of paranormal experiences, special connections to other dimensions of reality. These stories are appealing. They are genuinely appealing. Not because they are true, but because they take a real mystery and clothe it in significance. Cosmic, spiritual, mythological significance. They make the mystery feel meaningful in the way that only stories can make things feel meaningful. But let us be gentle and precise about what the evidence actually says. The Rh means we're not related to primates. Claim is based on a misunderstanding. The Rh factor is named for the rhesus monkey because antibodies produced from rhesus monkey blood were used in its initial characterization. But the Rh blood group system is present, in related forms, in all great apes, chimpanzees, gorillas, orangutans. It is a mammalian blood group system with a very ancient evolutionary history. Rh-negative people are not less primate in any meaningful genetic sense. They simply lack the expression of one specific protein within a blood group system that we share with our primate relatives. The alien hybrid hypothesis runs into the rather fundamental problem that Rh-negative blood shows a perfectly explicable geographic distribution consistent with normal human population genetics. If it were introduced by extraterrestrial interbreeding, we would expect to see it arising independently in multiple unrelated populations around the world at roughly similar times. Instead, we see it concentrated in specific, identifiably ancient lineages, exactly what you would expect from a variant that arose in one or a few ancestral populations and was subsequently inherited and distributed through normal human demographic history. The psychic abilities claims are not supported by any controlled, replicated scientific study. Anecdotal reports are abundant. Controlled evidence is absent. But, and this is important, many of the feelings that feed these stories are connecting to something real. The sense that Rh-negative people share something ancient and distinctive. True, they share a genetic inheritance that in some lineages reaches back tens of thousands of years to some of Europe's oldest human populations. The sense that Rh-negative blood creates biological difference. True, the absence of the Rhd protein has real, documented effects on immune function, pregnancy dynamics, and cell membrane structure. The sense that there is a mystery here that mainstream science has not resolved. True, the evolutionary paradox is genuine, and the full explanation remains incomplete. The myths are grabbing at something real. They are reaching for a genuine mystery and explaining it with the tools of story rather than the tools of science. And in the absence of complete scientific explanation, story rushes in. The task for those of us who love both truth and wonder is to hold the genuine mystery with as much reverence as the myths, while being honest about the limits of what we actually know. The real story, an ancient blood variant, a geographic distribution that maps on a deep European prehistory, an evolutionary paradox without complete resolution, is already extraordinary, already mysterious, already full of wonder and depth and significance. It does not need alien visitors to make it remarkable. It already is. Rest in that real wonder tonight. You are remarkable, not because of alien ancestry or supernatural gifts, but because of the actual, verifiable, extraordinary truth of what you are. The end point of a chain of human survival so improbable and so long that it reaches back to the Ice Age and beyond. That truth is enough. That truth is more than enough. We are near the end of our journey now. Not quite there yet. There's still a few more miles of dark to walk together, a few more scenes, a few more slow revelations. But we are in the later hours. The fire is burned lower, and the stars outside are at their brightest. That particular brilliance that comes only after midnight, when the dew has settled the dust and the atmosphere has achieved its clearest. So let us stop walking for a moment. Let's just sit, right here, and let me ask you something. What do you do with a mystery? Not a mystery that needs solving. Not a lock with a key. Not a puzzle with a solution. Not a question with a correct answer waiting in the back of the textbook. But a genuine mystery. An open one. A mystery that science is still circling from multiple directions, shining its various lights into the corners without yet illuminating the whole. What do you do with that? I think, honestly, you do what we're doing right now. You sit with it. You let it be large. You resist the urge, the very human, very understandable urge, to fill it with a quick answer. To collapse the mystery into something manageable and closed and satisfying. To say, it's aliens, or it's just random drift, or it doesn't matter, or the scientists will figure out eventually. Because the mystery, held open, is more valuable than any of those closures. The mystery is an invitation. An invitation to acknowledge that the world, this same world you inhabit, the one with its grocery stores and its commutes and its scrolling feeds and its manageable ordinary difficulties, is stranger and deeper and older than it usually appears. That beneath the surface of the ordinary, at every scale from the molecular to the geological, there are depths that have not been plumbed. Volumes of reality that remain, as yet, uncatalogued. In tonight's mystery, the Rh-negative enigma, this quiet biological question written in the blood of 1 in 7 people, is a particularly personal one, because it is not just a mystery about ancient humans in a landscape you can only imagine. It is a mystery about you, about your blood, right now in this moment, about the particular version of a very old question that is written in your genome, on chromosome 1, in the region where the RhD gene either is or is not. Whether you are Rh positive or Rh negative, you are part of this story. If you are Rh positive, you carry the ancient antigen, the protein that has been present in the great ape lineage for tens of millions of years. The standard bearer, the majority, the blood that Neanderthals had, that the Anatolian farmers who transformed European prehistory had, that most of the human population has and has had across the long millennia. You are the continuity. If you are Rh negative, you carry the absence, the deletion, the molecular trace of a change that happened somewhere in the deep past, in an ancestor who lived perhaps before the last ice age or during it, and has been passed forward with remarkable persistence across tens of thousands of years to find its way, finally, to you. You're the anomaly, the ancient echo, the 15%. And in either case, in both cases, you are carrying the record of everything that happened to get your particular configuration of molecules into existence. Every survival, every partnership, every child born in the cold to parents who managed, against all odds, to continue the chain. Your blood is not just a biological fluid. It is a document, a very old one, written in the most intimate possible language, the molecular language of proteins and genes and chromosomes, and addressed, across all those thousands of years, to the specific person who now occupies the body it runs through. To you, addressed to you. The letter reads, roughly, we were here. We survived. We passed this on to you. The cold was real, and the dark was real, and the loss was real, but we kept going. And here's the record of that keeping going, written in the only language that outlasts everything else. Let that settle. Let it move through you slowly, the way warmth moves through cold hands held near fire. You don't need to know your blood type to receive this letter. You don't need to understand the molecular biology or the population genetics or the evolutionary paradox. You just need to be here, breathing, alive, with the blood moving through you in its ancient, quiet way. You are the letter delivered. You are the chain still intact. And somewhere in the cells of your blood, whether they carry the RhD antigen or not, is the physical, measurable, verifiable record of every ancestor who lived long enough to love someone enough to create the next link. That is the existential truth at the heart of this mystery. Not whether Rh-negative blood is alien or divine or the mark of a lost civilization. Just this: You are here. Against extraordinary odds. You are here. And the blood in your veins knows the way home, because it has been making the journey for 300,000 years. Rest now. Let that truth be your blanket. Let it be warm enough. The laboratories are still lit. Right now. Tonight, as you rest in the warm dark, and these words move through your drowsy awareness like the slow current of a very deep river, there are researchers who are awake. Not metaphorically, literally awake, in the particular late-night way of scientists whose experiments don't pause for sleep, whose datasets are large enough to require weekend hours, whose questions are interesting enough to overcome fatigue. They're working on this, on the RH enigma, on its evolutionary biology, its ancient DNA signatures, its immunological implications, its potential connections to disease susceptibility. The mystery we have been living in tonight is not a closed case. It is an active investigation, with laboratories on multiple continents contributing to an international conversation that is slowly, carefully, accumulating new data. Let me tell you what the current frontiers look like. The ancient DNA front is the most active. Ancient DNA research has undergone a revolution in the last decade, driven by new laboratory techniques that can recover and sequence increasingly degraded genetic material from bones and teeth that have been in the ground for 10, 20, 30, 40,000 years. The sequencing of ancient human remains from key archaeological sites across Europe, the Middle East, and North Africa is filling in the demographic map of prehistoric human populations with a resolution that was unimaginable 20 years ago. Within this data, Rh status can often be determined by identifying the presence or absence of the RHD gene deletion characteristic of Rh-negative blood in European populations. As the ancient DNA database grows, researchers are assembling an increasingly precise timeline of when and where the Rh-negative variant appears in the archaeological record. The early results, as we noted in an earlier scene, are consistent with high Rh-negative frequency in pre-Neolithic Western European hunter-gatherers and lower frequency in incoming Neolithic farmers. But the dataset is still growing. The picture is still being sharpened. Within the next 5 to 10 years, ancient DNA research may be able to tell us with far greater precision when the Rh-negative variant arose, in which population, and how its frequency changed through the demographic upheavals of European prehistory. On the immunological front, researchers continue to investigate the broader biological consequences of Rh blood group status. Several ongoing studies are examining correlations between Rh blood group and susceptibility to specific infections, viral and bacterial, controlling carefully for other variables. The toxoplasma hypothesis, as we discussed, remains active, with new studies designed to test it more rigorously. There is also growing interest in the role of Rh proteins in the physiology of the nervous system. Recent research has found that Rh-like proteins exist not only in red blood cells, but in certain neurons and other cell types in the brain. The function of these non-erythroid Rh proteins is not fully understood, but their presence in the nervous system raises intriguing questions about whether the Rh blood group system has functions that extend beyond blood cell biology, and whether variations in Rh status might have subtle effects on neurological physiology. This is speculative at the current stage, but it is scientifically motivated speculation, the kind that leads to testable hypotheses rather than mere assertion. On the evolutionary front, population geneticists are applying sophisticated new mathematical models to the problem of how the Rh-negative variant has been maintained at its current frequency. These models try to determine what selection pressures, positive, negative, or balancing, will be necessary to produce the observed distribution. The mathematics is complex, and the results are, so far, consistent with several different hypotheses. The data does not yet cleanly distinguish between them, but the approaches are becoming more powerful. The combination of large ancient DNA datasets, modern whole genome sequence data from diverse global populations, and improved computational models for simulating evolutionary dynamics is producing progressively more refined estimates of when the Rh-negative variant arose, how quickly it spread, and what selection pressures it experienced. The answer, if it comes, will probably not be dramatic. It will probably not be a single, clean revelation. Rh negativity persists because of X, and X is Y. It will more likely be a gradual clearing of the picture, a slow movement from blurry to sharp, from ambiguous to resolved, in the incremental way that complex evolutionary questions tend to be answered, not with a single breakthrough, but with the accumulation of compatible evidence from multiple independent lines of inquiry. But the answer is coming, in the same way that the answer to hemolytic disease of the newborn came. Not immediately, not easily, but through the patient, cumulative work of many researchers over many years, each one adding a piece to a puzzle that they could only partly see. The mystery is being held, and the holding is being done with care. That is worth knowing, worth resting in. The world is full of people whose work is the careful, patient pursuit of true things. People who get up every morning and go to laboratories and field sites and computational desks and sit with hard questions without forcing easy answers. The RH enigma is in their hands. And in the meantime, in the beautiful, long meantime of scientific process, it is also in yours, in your blood, in your sleeping body, in the molecular record that no researcher has fully decoded yet, but that you are, in your own particular way, the living embodiment of. Let the labs work. Let the data accumulate. Let the picture slowly clear. You don't need to solve it tonight. Nobody does. We are very close to the end now. The fire is very low. The last good hour before dawn. That particular deep quiet where even the birds have not yet begun. The hour that belongs, completely, to the interior, to whatever is left of waking awareness when the busy mind is finally softened and the deeper, older knowing begins to surface. Stay with me just a little longer, because I want to give you something before we let the story go. Not a fact, not a date or a name or a scientific mechanism. Something different, something I've been building toward across the entire length of this night's journey, and which can only land properly now, in these late, quiet, almost sleeping hours. A perspective, an existential perspective that I want you to carry gently with you as you drift into sleep, and that I hope will still be there, soft and permanent, when you wake. Here it is. Every human being alive today is the direct, lineal descendant of an unbroken chain of reproductive success. Let that be specific. Let it be personal. Let it be yours. You, the person in the bed, the person whose breath is slower now than it was when we started, whose eyelids are heavier, whose thoughts have acquired that particular soft quality that precedes sleep. You are the product of a chain of reproduction that has never, not once, broken. From the moment our species emerged, approximately 300,000 years ago in Africa, every single person in your direct ancestral line survived long enough to reproduce. Not just survived, but survived specifically to the age at which they could partner with another person and produce a child who survived, who in turn survived, who produced another child who survived. Every link in the chain, unbroken. 300,000 years is approximately 12,000 generations. 12,000 people who stood between you and nothing. 12,000 people who were chased by predators and ran fast enough. Who got sick with things that should have killed them and recovered. Who found food when food was scarce. Who found partners when a band was small and the options were few. Who gave birth, or whose partners gave birth, in times before hospitals, before midwives with real training, before antibiotics, before any of the technologies that have made birth and childhood survival routine. They did it anyway. In the cold, in the dark, in the aftermath of volcanic winters that reduced the global human population to perhaps a few thousand individuals. In the crossing of the Sahara and the wading of shallow straits and the navigating of ancient coastlines. In the building of camps and landscapes that had never held a human camp before. Your RH blood type, whether positive or negative, is one small piece of what they carried forward. One molecular detail in an incomprehensibly complex inheritance. If you are RH negative, somewhere in that chain, perhaps 40 or 50,000 years ago, perhaps longer, one of your ancestors experienced the genetic event that silenced the RHD gene. A deletion, a change, and that ancestor survived, reproduced, passed the change forward, and the next generation survived, and the next, and the next, all the way to you. The blood that was an evolutionary question mark, the blood that, under certain circumstances, made pregnancy harder, persisted anyway, was carried forward anyway, by people who were strong enough, or lucky enough, or living in circumstances that minimize the cost, or perhaps experiencing some compensating benefit we have not yet fully characterized. 12,000 generations of anyway. 12,000 generations of, it was hard, and we did it anyway. And the reward, the staggering, impossible reward of all that persisting, is you. Alive, breathing, here. Whatever difficulties you carry into sleep tonight, whatever worries or regrets or old griefs or current anxieties fold themselves into the space behind your eyes as you begin to drift, bring them into the context of this chain. You are not the first person in this lineage to lie in the dark, uncertain about tomorrow. Not even close. You are not the first to feel the weight of life pressing down, the cold of circumstances beyond your control. Not even close. You are the inheritor of 12,000 experiences of that pressing weight and that cold, and 12,000 instances of surviving it. The capacity for survival is not just something your ancestors had. It is something they gave you. It is in your body, in your immune system, in your nervous system, in your capacity for connection, and your ability to fall asleep, even in difficulty, to let the night do its restorative work, to wake refreshed and continue. It is, in the literal sense, in your blood. The RH factor, positive or negative, present or absent, ancient or derived, is just one word in a very long letter. The letter that your ancestors wrote in the language of survival and addressed to you. You received it. You are reading it with your living body every moment of every day, whether you know it or not. Rest now. Let the chain hold you. It has been holding people just like you, cold, uncertain, sometimes frightened, sometimes lonely, always fundamentally strong, for 300,000 years. It will hold you through this night. It will hold you. The fire is embers now, just the warm red-orange glow of the deeply buried coals, pulsing very slowly, barely distinguishable from the dark around them. The kind of fire that keeps the cold away without asking you to watch it. The kind of warmth that requires nothing from you now except your presence. You have been very patient and very present, and I want to acknowledge that, gently, without ceremony, before these words find their end. You stayed with the blood, with the ancient question, through the mountains and the laboratories, the cave paintings and the chromosomes, the parasites and the Neanderthals, the folklore and the evolutionary paradoxes and the gentle, careful science that is still working, still patient, still adding its careful increment of understanding to a mystery that is 300,000 years old and shows no sign of resolving itself quickly. You stayed, and the staying itself was a kind of understanding, because the capacity to stay with a mystery without demanding that it resolve, to hold a question open with curiosity rather than anxiety, is one of the most remarkable and specifically human things there is. So let us in very slowly now. I'm going to bring you back to where we started. Back to your hand. Feel it where it rests, wherever it is, against the warmth of the blanket or the cool of the sheet or the familiar weight of your own chest rising and falling. Feel the blood there, warm, quiet, doing its ancient, continuous work without asking for your attention or your gratitude, moving through the small geography of your hand, through capillaries so fine they can carry only one blood cell at a time, in a circulation that has not stopped for as long as you have been alive, carrying oxygen to the waiting tissues, carrying carbon dioxide back, carrying immune cells and platelets and hormones, and the 100 other passengers of the blood that maintain the living equilibrium of your body, moment by moment, through the night, carrying, on the surface of those red blood cells, whatever molecular flag it carries. The RHD protein, perhaps, expressed in the amber language of the ancient antigen, connecting you to the Anatolian farmers and the great ape lineage and the long, continuous history of that protein's presence in the mammalian story. Or the silence, the clean, smooth absence that is RH negativity. The deletion, the inheritance of an ancient Western European hunter-gatherer whose name we do not know, whose face we cannot reconstruct, whose life lasted perhaps 30 or 40 years in a cold and beautiful and dangerous landscape, and whose small genetic change has been riding forward in the blood of that lineage's descendants for, we believe, somewhere between 30 and 60,000 years, to arrive at your hand, right now, tonight, The names don't matter now. RHD and chromosome 1 and haplogroups and founder effects and ancient DNA and hemolytic disease and Rh immunoglobulin and the Basque language and the Atlas Mountains and the Neanderthal genome. All of it can soften now, can blur at the edges, can dissolve into the warm, general sense of having been somewhere important and having returned safely, like footsteps in the sand. As the tide comes in, they were there. They were real. They told a story. And the tide is coming in now, slow, patient, inevitable, kind, and the details will soften and the specifics will blur, and what will remain is not the names but the feeling of the story. The feeling of being small and connected at the same time. The feeling of being part of something that reaches further back than you can see. The feeling of being the recipient of an inheritance you did not earn and cannot fully understand, but which is undeniably, unambiguously, completely yours. You are the current end of a 300,000-year story. You are where the blood arrived. You are the living address of a very old letter. And the letter says, you are enough. You are always enough. The chain required you to be exactly what you are, and so that is what you became. The blood that reaches you is the blood that survived everything it met, and it lives in you now, warm and quiet, keeping its ancient rhythms through the night. The embers are very low. The stars outside are beginning their slow seasonal drift toward the horizon, making their enormous circuits, utterly indifferent to the small warm lights of living things below. But we are not utterly indifferent to each other. That is the other thing the chain produced. The care, the curiosity, the gathering together around a fire, real or metaphorical, to tell the old stories, to ask the old questions, to sit with mystery without demanding resolution, to accompany each other through the long, dark, beautiful hours of the night. Right now, in Tokyo and Lagos and Lima and Oslo and Istanbul and the Welsh valleys and the Atlas Mountains and wherever else in the world people are lying in the warm dark and letting these words carry them towards sleep. Right now, we are all doing this together, in the same dark, under the same stars, each carrying our own particular version of the ancient inheritance, each the end of an unbroken chain, each, in some small molecular way, still connected to the first fire. Let your breathing slow all the way now. Let your body grow heavy in the best possible way, the way of completed journeys, of good tiredness, of the particular peace that follows being genuinely held by something larger than yourself. The mystery will be here when you wake. The science will have moved forward by some small increment. The ancient blood will still be ancient, and you will still be here, still the end of the chain, still the recipient of the letter, still, in your blood and your breath and your sleeping and your waking, the living proof that something very old decided to keep going, all the way here, all the way to you. Good night. Sleep well. You are held.